Alexandra Dulebo, and I'm a biocells application engineer at Bruker. And I would like to welcome you to this installment of the Bruker AFM webinar series. This presentation is titled High-Resolution Imaging of Chemical and Biological Sites using PeakForce QNM Atomic Force Microscopy. It will be presented by me together with our guests, Dr. David Elstins and Moritz Freundschur from ETH Zurich at Department of Biosystems, Science and Engineering based in Basel in Switzerland. Before we will start, I would like to make a few quick logistical comments. First, we encourage your participation during the webinar. If you have a question, please type it into the questions dialog box on your screen. We will accumulate these questions through the presentation, group them together, and then answer them at the end of the prepared remarks. Quite often we have more questions that we can answer in the session, and in those cases we will be sure to follow up with the answers via email afterwards. Also, if you would like to review the presentation afterwards or pass it on to a colleague, the webinar will be posted online within 1 week in the webinar section of the Bruker web page. As a follow-up to this presentation, you will also be receiving an email with this link. Finally, when you exit the webinar software, you will be asked to take a brief survey. We would very much appreciate it if you would take the time to complete this, as it helps us pick the topics that are most important to you and to generally make this series better. So let's get started. Today's webinar is dedicated to rapidly growing need to identify and quantify the molecular interactions, both with high force sensitivity and high lateral resolution. In this webinar, I will introduce you Bruker's advanced technology that, in combination with chemically functionalized probes, could be used to simultaneously contour the surface and map biophysical properties of biological samples at the nanoscale. After my introduction, I will pass the word to our guests who will present how they utilize this technology in their research. Going directly to the core of the topic, we will be talking about molecular recognition imaging, where chemically modified cantilever tip is used to acquire simultaneously the topography of the sample, as well as map specific adhesion or recognition between the tip and the sample. On this slide, you can see the surface of a live but malaria-infected human red blood cells that possesses knob-like structures on their surface. This knob seems to play a crucial role in attachment of the infected cells inside the blood vessels. Scanning these cells with the probe chemically functionalized with the receptor molecules similar to those present on the endothelial cells lining our blood vessels lead to a positive signal on the adhesion map. Overlaying topography and adhesion nicely represent the binding spots for receptors on the cantilever tip are located only on the knob-like structures of the cells. So let me now tell you about the technology that enables such detection, as well as other advantages and type of information you can acquire using it. This AFM-based technique is Bruker's recently released AFM imaging mode called PeakForce tapping. In PeakForce tapping, the atomic force microscope probe is modulated at low frequency, about 1 or 2 kHz, using very small amplitudes, typically 100 nanometers or less. As the probe is brought in contact with the sample surface, the feedback signal is the maximum force, or so-called peak force, between the tip and the sample. So if you think about the motion of the probe in terms of z position, we are essentially performing a triggered force curve at every position on the sample surface. While this may sound very familiar and similar to force volume imaging, there are some very key differences in the operation of these two modes. In PeakForce tapping, we're using sinusoidal ramping and not linear ramping as it used for force volume or single force curve measurements, also known as force spectroscopy. With each interaction of the AFM tip with the sample surface, the feedback loop is continuously detecting the peak force. As such, the imaging force control benefits from the results of the previous tip-sample interactions. Together with sinusoidal ramping, wherein the tip velocity approaches zero as the tip approaches the surface, These enables PeakForce Tapin to directly control the tip-sample interaction. Force therapy facilitating the use of ultra-low set points such that imaging forces can be easily kept below 50 piconewton, even when operating in fluid environment. This helps protect both the AFM probe and the sample from potential damage and is really the key to enabling high-resolution imaging. Additionally, force volume and single force ramps are typically performed at frequencies of about 1 to 10 Hz, requiring significant amounts of time to acquire data. Because PeakForce Tapin mode operates at higher frequencies, usually 1 or 2 kilohertz, we are essentially performing thousands of force curves per second. This allows PeakForce Tapin images to be acquired at traditional imaging mode scan rates and at a high pixel resolution. And since there is a tip-sample interaction or force distance curve behind every pixel of collected image, we can extract such information like elastic modulus, deformation, and of course the adhesion forces between the tip and the sample. With respect to the topic of our today's webinar, when using chemically modified probes, the unbinding forces between the molecule or chemical group and the tip and molecule or chemical group on the surface could be measured. So when it comes to imaging biological samples, AFM has an important advantage of being able to operate in fluid, so that samples can be observed under more physiologically relevant conditions. And with PeakForce Tapin, not only do the low imaging forces protect soft biomolecules and cells from being distorted or damaged, PeakForce Tapin also makes imaging in fluid much easier to do. First of all, there is no requirement for use of specialized probes. Even colloidal probes could be used. And with the eliminating of the need to tune the cantilever resonance, there is no longer a worry about peak shifting. PeakForce Tapin technology has also facilitated scan assist mode, which is the self-optimizing imaging mode. Auto-optimization of the imaging set point prevents set point drift, as often happens in traditional Tapin mode operation in fluid due to amplitude dropping over time. Together with auto-optimization of other parameters such as gain and scan rate, PeakForce Tapin mode results in faster, more consistent data regardless of the user skill and level. With PeakForce Tapin and scan assist modes, the ability to acquire high-resolution images such as that shown here of the single herpes simplex virus is more consistent and no longer heavily dependent on operator experience. All that makes PeakForce Tapin mode a perfect solution for molecular recognition measurements, where interaction forces between two molecules or chemical groups, one which is attached to AFM tip and other bound to the surface, are probed. First, the tip is brought down to the surface until it contacts and exerts a positive load on surface. Then the tip is retracted and moves back to its original position. During this retraction, a downward peak may occur in the retraction curve that indicates adhesion between the tip and the sample. If the spring constant and the deflection sensitivity of the cantilever is known, the tip sample and binding forces can be measured. And when the tip is functionalized for molecular recognition measurements, one challenge becomes the ability to distinguish between non-specific interaction and specific interaction. For this reason, it is common to use intermediate molecules, also called spacers or linkers, between the tip and the molecule. The greater flexibility of linker provides the ligand molecule some mobility to best assess the binding receptor. It also introduces a characteristic curved adhesion peak due to entropic stretching of the linker molecule, which helps distinguish the desired specific interaction from a non-specific interaction. On this animation, you can see the characteristic force curve with the specific interaction where the linker is used. For common approaches to tip functionalization, you can browse Bruker's application note number 130, which you can find on our Nanoscale World Forum. There you will be able to find many other application notes on different topics and different AFM modes and techniques, recordings of the previous webinars, and you can also post your own AFM-related questions to us and to the AFM community. I would like to say a few words about software improvements that have been introduced recently to make recognition imaging using PeakForce tapping even better. In the latest release of this 64-bit version of the nanoscope software, which is 9.1, several adhesion algorithms have been introduced. One of them, which is called section minimum, enables user to define the part of the retract curve used for adhesion calculation in real time. It will be particularly useful for those who are functionalizing their probes using flexible linkers, spacers, to distinguish specific adhesion and provide more degrees of freedom to their molecules. And in the end of my part, I would like to share with you the number of publications being published using Bruker's PeakForce tapping technology, with more than 340 papers up to date in various fields, ranging from life science and nanomaterials down to polymers and electrical characterization. We see that majority, or 75% of people, uses the PeakForce tapping technology for its unique data type it produces. Another thing we find quite important is that more than 30% of publications from our users are actually published in high-ranked journals with the impact factor above 4. That is particularly encouraging if we consider that only 10% of all publications published nowadays are entering such high impact factor journals. With that, I would like to finish my part and pass words to our guests and introduce them to you. So the first speaker will be Dr. David Elstins, who is now working at ETH Zurich at Department of Biosystems Science and Engineering based in Basel. The head of the group is Professor Daniel Müller. Daniel did his master and PhD at the Catholic University in Louvain in Belgium in nanobiotechnology with the Professor Yves Dufresne. David's research focus at the moment in quantifying interactions of single molecules on molecular and cellular level. And David is the co-author of 45 publications and 6 books. After David, we will hear Moritz Freinschur, who is permanently doing his PhD at the same place at ETH Zurich. Moritz performed his undergraduate studies of chemistry and biochemistry at the University of Freiburg in Germany and at the Concordia University in Montreal in Canada. Moritz's research is based around high-resolution imaging of single native proteins coupled with the quantification of biochemical and physical properties using functionalized AFM tips. Moritz is a co-author of 4 publications in that field of research. So, please.
Hi everybody. So I'm David Elstins, and I will try to talk during 15 minutes about the use of PeakForce tapping to map chemical and biological interaction on live cells and biological samples. So originally the AFM was invited as a topographic tool, and it rapidly evolved into multifunctional tools, enable us to map chemical interaction or mapping single cell surface receptor or measuring the energy landscape of individual receptor-ligand interaction. To be able to detect some specific or unspecific interaction, we need to use a functionalized tip, and so the AFM is like a lab on the tip. There is different different range of chemistry that we can use to map specific and unspecific interaction, and I will mainly focus on 2 kinds of chemistry. The first one is the use of thiol and gold. So for this, we need to use a gold tip. So this is in yellow, and we can just evaporate some gold layer on the silicon tips. And after, you just immerse the tip overnight in alkaline solution, and by using this kind of chemistry, you can, for example, have hydrophobic tips or put some protein covalently attached to the tip using a mixture of COH alkantyl and OH alkantyl. so the protein is covalently anchored on the tip. Also, if you have the possibility to have an histide protein, you can use a mixture of NTA alkyl thiol and polyethylene glycol, which prevent from unspecific adhesion. So this is for the thiol angle. Otherwise, you can also use different kind of chemistry, and the use of linker, which able to more define the specific and specific adhesion, because this spacer can prevent from the non-specific adhesion, so you will have no peak in the contact area of the force curve. So in this kind of chemistry, you first add amino group on the tip, and after you react with the cross-linker, and finally you graft the protein on the added group function on the tip. So as I told you, the AFM was at the first step a topographic tool, and it rapidly evolved into a multifunctional tool. And at the beginning, people tried to measure single interaction between a receptor and a ligand, and Florin et al. In 1994 first measured the interaction between navidine and streptavidin, here in this paper, and are able to measure single interaction or multiple interaction at the same time. Several years later, the tool evolved to an affinity mapping imaging tool. So to do that, for example, in this paper, Grombois et al. Use a functionalized tip with some lectin to map a confluent layer of red blood cells. So it's a mixture of group A and group O red blood cells, and they use specific lectin against the group A red blood cells, and they are able to distinguish the red blood cells with from the group A in opposition to the group O. So it's the first paper who showed this kind of mapping. And recently we show also that we are able to follow dynamic processes. In this paper we are able to localize individual adhesion on the Candida albicans yeast. So here you have a yeast trapped into a polycarbonate membrane, and we are able to localize individual protein and also follow the organization of this protein across the time. So this is very good to follow dynamic processes, but we see in this paper that we reached the limitation in terms of resolution and temporal resolution. So we try to find another way to have better resolution from the point of view of the lateral resolution and the temporal resolution. And so more or less at the same times, the AFM-based, the force distance-based AFM technology appeared, so like the PeakForce tapping from Bruker. And in these methods, the tip is associated on the surface with a speed of 3 order of magnitude faster. And so we have a much better temporal resolution, and also we can register more pixels, so which can be very good to follow dynamic processes. And from this kind of mode, we can extract all the curve from each pixels, and we can extract the parameter like the adhesion in which we are interested. But the question at this time was, is this method quantitative and able to map chemical and biological sites with high resolution? So to answer this question, we first try to find a way as a proof of concept, and we decide to use organic substrate, which are here round shape hydrophilic pattern. So it's a silicon oxide round shape form, and these are surrounded by hydrophobic silane. We first use hydrophobic tip in conventional force volume with a speed of 1 Hertz, and we acquire 64 by 64 maps. So this kind of maps takes around 1 hour to be recorded, and we can extract the parameter. So we found that an adhesion force of 7 nanonewton on the silane and around 2 nanonewton on the silicon. We next do the same experiment with the PeakForce technology, and we found, so in this case, the topography and the adhesion image are recorded at the same times, and it takes around 10 minutes to be recorded for 512 by 512 maps. And we found that the adhesion value for the silane and the silicon oxide are quite very similar, around 7 and 2 for respectively the silane and the silicon. So which means that this method is very quantitative, and we can trust the adhesion value that we acquire. As a control, we use an hydrophilic tip, and we measure nearly no adhesion, neither on the silicon or the saline. So from this first proof of concept, we show that we increase the lateral resolution, we have a better temporal resolution, and we can maintain the quantitative information as true. We next try to go to a more complex sample, and we move to the Aspergillus fumigatus cells, which is human opportunistic pathogen. And in this case, we try to map the hydrophobicity of the cell surface. So the cell surface of this kind of spore, of fungi, is made of hydrophobin. It's a very hydrophobic protein, which is organized in rodlets. And upon germination, this rodlet progressively disrupt, and the underlying polysaccharide layer become exposed to the cell surface. So we put the cells for germination for 2 hours, and so we expect to have a mixture between some rodlets and some underlying polysaccharide. And here you have the topography imaging and the adhesion channel, which is recorded at the same times. And you can see that in the area where you see the rodlets, we see a very high adhesion, so which means that we are able to detect the hydrophobic protein, rather than in the part where you have the amorphous material, you have no adhesion. These kind of measurements are in good correlation with previous measurements that we published several years before. And so we can again say, okay, this kind of method is able to detect the chemical properties of individual cells directly on live cells with a very high quantitative measurements. Finally, as a proof of concept, we try to map the localization of individual protein on live cells. And we use an NTA tip. So we just use alkyl terminated by NTA groups to detect, in fact, individual protein level within this tag. So this is a sensor. So it's a transmembrane protein who detects the cell surface stresses, and which can go inside the cells to modify the cell cycle, for example. Here you have an image of the yeast cells, which is trapped in the polycarbonate membrane, and you have a higher resolution imaging, and at the same time, this is the adhesion channel where you see the green spot as a single interaction. So this is the kind of curve that you can extract from the PeakForce image, and you see a single peak with an adhesion value around 300 piconewton, which is the good value for histag NTA interaction as such loading rate. And we compare the maps with the map that we can obtain in conventional force volume, and also the force curve that you can obtain here. And you see that you have more or less the same kind of force curve. The adhesion value is a little bit lower in conventional force volume because the speed is much smaller. If you look at higher resolution, you can see on the higher map that you have small pixels which are around 4 nanometer in lateral resolution. So we can see that more or less we have a resolution of 10 to 20 nanometers on live cells. And we assess the specificity of this interaction by using the same tips but without the nickel in solution. And here we see that we have a large decrease of the interaction, which means that the interaction that we measure are specific. We also show that on the bud scale, where the cell wall is thinner, we have much more adhesion, which is due to the fact that the protein are more accessible to the tip. If you can compare the resolution in PeakForce with the resolution in conventional force volume, you see that if you would like to say something about the organization, is very difficult here in conventional force volume because you have only a few pixels and you don't see nearly nothing. Rather that in PeakForce, you can see that, for example, here the protein seems to be organized like a network with some space where the protein are not. And we believe that this organization reflects the organization of the protein in the plasma membrane, where the sensors are excluded from nanodomain. And so we can start to say something about the organization. So in the next study, we start to use this technology to solve more more complex biological question. And for the small story, I already tried to study this kind of experiment at the beginning of my PhD with the conventional force volume, but I was not able to have any results because we have only a few adhesion on very localized place on the bacteria. So you will see this kind of measurement was not possible before. So in this study, we try to map the extrusion site of a single bacteriophage from the bacteria. So this is a filamentous bacteriophage, which is a virus who infects gram-negative bacteria. This phage consists of a single-strand DNA, and there is some protein who assemble around this DNA. So you have like 9 proteins. Some protein are for the virus assembly, and some other protein insert into the cell wall to make like a channel so that the virus can go out from the cells. We had at the terminus of the virus some E-tag so that we are able to detect it with a functionalized tip with an TA group. Here you have 2 different electron microscopy image. In the first one, the cells here, the Escherichia cells are infected with wild-type phage, and you can see like small air around the cells. But you are not able to distinguish from where are the virus, and it's very difficult to say where are localized the extrusion sites. If you use a mutant of the virus without the protein 3 here, the virus are not able to escape from the cell wall because at the end of the assembly, this protein is missing, and finally the virus is not able to go out. And so finally we have a bacteria with a lot of air, which are the virus, which are very long because they add some DNA more and more and more, and finally you can have bacteriophage up to several micrometer. As a first proof that the NTA is well on the phage, we use fluorescence microscopy to assess where is the E-tag group. And so we had a dye against the E-tag green dye. And you see with the wild-type phage that the pole of the bacteria and also the septum, which is not shown here, are colored in green. For the mutant phage from the P3, you will see that there is a high expression of the phage because they cannot escape from the cells, and these are localized everywhere. As a control, we also use a cell which is not infected by the phage, and we have no fluorescence. So we use the PeakForce technology to map the E-tag on the cells, which means the extruding bacteriophage. So here you have dividing bacteria. Here is the septum region, so it's a higher resolution. And on the adhesion map, you will see that here you have some adhesion event. Resolution, you see these kind of maps. And we assess the specificity by adding some ADTR to the solution, and we see a very huge decrease of the adhesion frequency. Here on the edge of the cells, there is some adhesion, but it's very difficult because if you look at the individual force curve, they are not well defined. It's very difficult to see if it's real adhesion or some noise or whatever. So, but we only analyze the force curve on the top of the cells, and this typical force curve you can have. So it's always adhesion around 350 piconewton, which is good value expected for this kind of interaction. And we tried in the second step to correlate the adhesion with different parameters that you can extract from the PeakForce files. And for example, we look at the mechanical properties of the cells, and we extract the Young modulus of the cell wall of the bacteria. And here on this kind of cells, you can see, for example, in the first area, that you have some adhesion even localized near the septum, and at the same time, you see that these adhesion even are localized in softer area, which are surrounded by stiffer material. You have also this in the second zoom here, which is more clear. You have a softer area surrounded by stiffer material. And in fact, we believe that this comes from the fact that some protein have to be inserted in, I can show you it here, inside the cell wall this kind of protein, and in fact the phage doesn't encode for any enzyme. So to insert this protein, we have to disrupt the peptidoglycan, and this cannot be done after the peptidoglycan assembly. So it has to be done directly when the peptidoglycan is synthesized, so directly during the division of the cells. And so it's why this kind of adhesion even are always localized near the septum. And because of the insertion of this protein in the cell wall, the assembly of the peptidoglycan is perturbated, and there is accumulation of newly formed peptidoglycan around these adhesive nanodomains. As a control, we also analyze the mutant phage, so the cells infected by the mutant phage, and we record adhesion nearly everywhere, so which is in good agreement with the fluorescence image. And at the same time, we see that the Young modulus image is very complex, because we think that all these inserted channel are perturbating the Young modulus and the assembly of the peptidoglycan. On the cells non-infected by the phage, you have no adhesion and nearly homogeneous distribution of the Young modulus across the cells. So which means that using this kind of PeakForce technology, we are able to solve a very more complex biological question, and this is very helpful to understand several processes. So now I will give the talk to Moritz.
Okay, so hello from my side, and I will continue now with the second part of this presentation. So what you've heard from David so far was some interesting studies on exploiting the PeakForce QNM mode to investigate ligand receptor pair interactions on kind of a large scale, considering the biological samples. So what we want to do now is we want to advance one step further, and we want to increase the lateral resolution of the receptor to be investigated, and of course also increase the resolution of the affinity map that we want to measure. So what you see here is basically a desired result. You can see a high resolution topography of a native protein, and on top of that you have a specific adhesion map which is indicating you the single receptor sites on this native protein. For this studies, we again started with a nickel-NTA his-tag interaction system. So again, as you have seen from the introduction of David, we are using gold-modified tips, gold-coated tips, which you can modify with thiols bearing either a functional group in the end, which is the NTA group, or only bearing a hydroxyl group in the end, which is giving you a non-specific interaction. Secondly, we want to have kind of a model substrate in order to test whether our nickel-NTA his-tag system is really working. Also, is it really working with the PeakForce QNM AFM mode? So what we start here with is a gold-coated surface. It's basically a freshly cleaved mica, which is gold-coated by electron beam evaporation. And afterwards, you're covalently binding peptides to this gold surface, which are bearing a terminal cysteine. So the specific peptide, it's a nonapeptide, which will have 6 histidines in the end. And the other peptide, which is an unspecific peptide, just has a random sequence of amino acid in the end. So when we now come to the application of our QNM PeakForce AFM mode, we need to think about what parameters can influence our ligand-receptor interaction mapping. And the 3 biggest parameters that we can change here is the contact force, also imaging force, the oscillation amplitude, as well as the oscillation frequency. because as you have seen in the introduction, the piezo actuator is oscillated in a sinusoidal way at frequencies between quarter kilohertz and now even 8 kilohertz. And what you can change is the amplitude of this frequency, and of course also the contact force that you apply when tip and sample are into contact. So if you look at one of these force curves and zoom into them, you will see that the contact area between tip and sample is this highlighted region, which is about a quarter or a third of the whole up and down movement. And now this contact area, which is here also indicated in red as the contact time, this can be mainly influenced by the oscillation frequency of the piezo actuator. And if we think about detecting specific ligand-receptor interactions, we need to think about the contact time between the ligand and the receptor, because basically for different ligand-receptor pairs you have different association and dissociation kinetics. So if you have a really high affinity ligand-receptor interaction, they will associate in a quite short time. You don't need too much of a contact time, but if it's weak interaction, you need a longer contact time to detect these ligand-receptor interactions. So what we have seen is that by modulating the frequency of this piezo actuator, we of course modulate the contact time between the tip and the sample. So this is our main parameter now to change contact times. Of course, amplitude and contact force also change a little bit the contact times, but our easiest parameter to change is in this case the frequency. So when we start at 2 kilohertz, we get rather short contact times between 30 and 70 microns, but we can modulate these contact times in one order of magnitude. If you go to a quarter kilohertz, we will reach 240 to 480 microseconds, which is in the biological range of a ligand-receptor formation. So for this model system that we have, a gold-coated surface with a HISTAC and a modified AFM probe with a nickel NTA group, what we can see immediately is that if we measure at 2 kilohertz, which is giving us quite a short contact time, we're only seeing adhesion forces which range from 0 to about 70 piconewton. So these can be related to unspecific adhesions, which are just due to the contact between the tip surface and the support. If you go for lower oscillation frequencies, we increase the contact time, and we see already that we have increased adhesion forces. So this is the first indication that suddenly our specific interaction is available due to the longer contact times. A second test is, of course, to screen these specific interaction. If we measure with the nickel in the solution and with the nickel not being in the solution, we see immediately that we are again reduced to non-specific interaction forces. So we can filter all these curves, collect them, and you will see there's a large range of rupture forces that we can measure, reaching from about 100 to 300 piconewtons. The rupture length is quite homogeneous because this short pack link here can't be stretched very much, and you will have a collection of these rupture forces which all have the same rupture event, as you can see here. So basically this system now tells us that we can use this modification, the gold modification, and we can use the parameters for the QNM AFM mode, which are namely a half 0.5 kilohertz of the oscillation frequency, and in this case about 30 nanometers of the amplitude. And using imaging forces of about 100 piconewton, we can detect a specific HISTAC nickel NTA interaction. And the positive force curves will look like this. So now we want to work on the native protein. So we really want to go and see what kind of resolution can we receive with a modified tip, and we want to see if we can really resolve single binding sites between the histag and the nickel NTA group. So the model protein that we have here is a so-called spindle assembly abnormal protein 6 homologue, SA6. It's important for centriole stabilization and dimerization. So it's basically a cartwheel protein which is surrounded by microtubuli. And as you can see here, it's a ring-shaped protein which has some 6 to 9 spikes around it. And what is so special about this protein is that it has some engineered histags at the C-terminal distal parts of these spikes. And this is really useful for us because we know where we expect our specific interaction. So we don't want to see any specific interaction in the main part of the protein, and if the experiment works, we only want to see specific interactions right at the distal part of these spikes. So what you see here is an overview of these native proteins now recorded with a normal tip, not modified. And this is a zoom in, and you can see this nice topography of this protein. So now we work with a nickel NTA functionalized tip. What you see at first is that the resolution drops a little bit. This is quite normal because you increase the tip apex due to the gold coating and the chemical modification. And what we can record now is an adhesion map which shows us the specific adhesion forces which happen when the nickel NTA group is interacting with the histag. So the adhesion forces are filtered, they're ranging from 100 to 200 piconewton, and you can see that they're correlated with the C-terminal parts of this ring-shaped protein. With the QNM PeakForce AFM mode, we can of course go into each pixel and select it and look at the force curve specifically. As you can see here, encircled in a red circle, you see these specific interaction force curves which have kind of a tether and a rupture event. Sometimes you also detect interaction forces which reach the events of 100 piconewton. However, if you look at them closely, you see that first of all they are not structurally related with the protein. Secondly, they appear in the tip sample contact region. So they can be called a non-specific interaction. Here I want to mention that the nanoscope analysis software comes along with a MATLAB utility box called NS MATLAB Utilities, and this enables you to import these PeakForce capture data files into MATLAB and process them. So we use this tool to simplify the work to create these adhesion maps. So what we could do here is we could import these files, load up the topography of the image, calculate an adhesion map, select the area that we want, show them, filter them according to our criteria, like the minimal adhesion force or the position where this adhesion peak should appear. And then we can screen through each single curve, and then we can save the curves, process them, or use them for our further analysis. As a test experiment, of course, we also did the same experiment without having the nickel in the solution. As you can see, there are some adhesion events. Again, these events are non-specific interactions. They appear in the tip sample contact region, and they are not correlated to any structural features of the protein. As you might have noticed, we didn't record all of the histag interaction sites in the protein. This can have manifold reasons. One of them, of course, is that the contact time is not enough yet. So it can be that the specific interaction between the ligand and receptor hasn't been established yet due to translational and rotational conformations of the ligand. So it didn't really find the histag. Secondly, could be that these histags of the native protein are facing the mica, so they're not approachable. And this is why we just decided to image this, this native protein several times, and then we would look what kind of distribution of interaction events do we have. And this is, sorry. So this is what you see here. We were imaging this area 3 times, and so now you're combining blue, magenta, and green interaction events. And what you see in the end is, if you overlay all of these events, is that about 80% of these interaction sites are successfully mapped. So in the end, it's a question of a probability whether ligand and receptor will be matched, and you will detect this specific interaction. As a statistical result, you can say you can plot all the rupture forces that we detected, which are again ranging from 80 to 220 piconewton. We'll have a wider spread of the rupture length here because most probably these flexible arms of these proteins will detach a little bit of the surface. This is what we will also see if we image this protein at a higher force, let's say 120 piconewton. We will simply disrupt the monomers. And here you can see all the collected specific interaction force curves. So concluding, we can say that we could use the PeakForce QNM AFM method successfully as a quantitative tool to image chemical and biological sites. Topography and adhesion can be directly correlated. If we work on large-scale biological samples like live cells, whether they are eukaryotic or prokaryotic, we can get a lateral resolution of about 10 nanometers and a way improved temporal resolution compared to force volume AFM. We can solve biological problems by correlating the adhesion maps with multi-parametric information like the modulus, for example. We can use this tip also to increase or to work on native single proteins and to increase the resolution, the lateral resolution, so we can get high resolution images and high resolution specific adhesion maps. A useful tool is the NSM MATLAB toolbox, which facilitates the data processing and analysis. And in this case, we could detect specific interactions on these native proteins to a size of about 3 nanometers. This brings me to the acknowledgements. In the left upper corner, of course, you see our group in Basel, the group of Professor Daniel Müller, the professor of David, Professor Yves Dufresne, at his former lab. And of course, we thank to all our collaborators, Professor Heinisch, Latge, Saumillon, and Steinmetz. And of course, we thank all the attendees for staying with us and hopefully listening attentively to us. Thank you.
David, Moritz, thank you very much for your nice presentation. And now we have some time for the question session. So let me start with the questions. The first one, what type of probes do you use to measure interaction forces among proteins and biological materials?
In my case, I was using sensitive probes, which are from Bruker, they're called BioLevers, with a spring constant of 0.07. If the same name is ACS40, if they're from the company of Olympus.
For the cells, we use Scanasys fluid that we coat with gold layer, and also the OTF4 from Olympus.
Okay. Then there is one question referring to Maury's presentation on the page 21, where you're showing the differences in the contact time with the different frequencies, PeakForce tapping frequencies. You are showing also the range. So the question is, yeah, the contact time in microseconds. So what's the reason for this range? Could be.
The reason for this range is that we also modified contact force and amplitude. So the amplitudes that we normally use for high resolution imaging is between 10 nanometers and 40 nanometers. And if you use a higher amplitude, of course, at the same frequency, the tip is moving faster, so the contact time will be reduced. So for example, this 30 microseconds will be at a higher amplitude and 70 microseconds will be at the lower amplitude.
Okay, thank you. Another question, can you please tell me what, in your opinion, would be the optimal chain length for the PEG spacer to be used for functionalization of the tips in combination with QNM?
In reference to high resolution imaging or to cellular imaging?
Well, you can mention both of them. I think it could be interesting to our audience.
So I would say for high resolution imaging you will try to have a linker as short as possible, but also as long as possible to identify specific interactions. So I guess the length would be about 15 nanometers.
Lower, I think. For high resolution, you say?
Yeah.
I think for high resolution, in fact, here we don't have any linker.
Yeah, I mean,
to the Alcantial, there is no very true linker. It's just 3 units.
Exactly, like for.
So we try to avoid such a linker because otherwise we lose a lateral resolution. And for cellular levels, yeah, it's dependent. I don't think that's the linker is so important because finally all the stretching comes from directly the cellular protein. But I think it's just very important to have a diluted protein or a group on the tip, and also ethylene glycol matrix to prevent unspecific adhesion on the tip. This is very important. After, the length of the polyethylene glycol is not so important. In some assays that we are performing now, it's very short linker. It's only less than 20 ethylene glycol units on linker.
All right. From my experience, I could also add that typical values like 4, 6 to 20 nanometers are used. But of course, you would like to keep that as small as possible when you're dealing with the smaller molecules. So the next question is, does the surface roughness affect the force profile? Do we need a flat surface to obtain the data like you showed us today?
Well, I mean, basically, it depends on how fast the system reacts to the changes in the topography. I mean, in my case, I can say we are scanning at half a kilohertz. This means I'm scanning really at a low scanning rate. So this kind of gives me not big troubles of overcoming big topographical obstacles.
For cellular levels, it can be a problem because, yeah, cells are not flat at all, and if the tip comes between different cells, in fact, the drag force can be very different, and finally, the force curves are very different on the top of the cells and between two cells, and the drag force play a quite huge role between cells, and in this case, some hysteresis can be taken as adhesion, in fact, by the software. So it's very difficult in this case to try to find a good probe that avoids too many drag force, and after, yeah, we need to use an offline software to remove this drag force to avoid that it's displayed like an adhesion event.
Okay. In general, as the conclusion, I would say that atomically flat surfaces is not required in that case to get the force profile. Very interesting question we have here. what would be the limit in frequency that would still yield sufficient interaction between ligand and receptor? And that's, I think, quite relevant to your slide, Moritz, which is open right now.
Mm-hmm. Yeah, as I said, like if I was working now in the regime of half a kilohertz, we're also working at a quarter kilohertz. If you go to 1 kilohertz, you need to work with receptor ligand interactions that have quite a low activation energy barrier, or let's say quite a low dissociation constant. So there must be really stable ligand receptor pairs. But so far I prefer to work at half kilohertz. This takes about 20 minutes per one image. This can be quite a long time, but I made the experience that it led to much better results in terms of detecting specific interactions.
But as you also mentioned during your talk that with the larger high frequencies there is a less adhesion forces or unspecific interaction that you recognize.
Mm-hmm.
Is it true? Yeah.
That's true. Because generally contact time is diminished. The question is whether there's still enough time to build up a specific ligand-receptor interaction. So we can't fool the timescales of biology by just scanning faster.
Right. Another relevant question regarding the scan rate, which you typically use for this type of measurement.
What do you use now?
The scan rate is, we try, in my case I try to go not so fast, and it depends also the frequency that we use, but the scan rate is nearly about 0.1 to 0.2 Hertz. I don't know for you, Matt.
Yeah, I think at half a kilohertz you can go to also 0.25 Hertz scanning rate. Somehow you need double as much time than you should need for the same pixel resolution if you record the PeakForce map. This is, probably Sasha, you're more familiar to that, why exactly you have to scan a little bit slower if you record all the PeakForce curves.
Right. So the answer is that you, in the high resolution imaging, so you've done, Moritz, you can scan a little bit faster, like you mentioned, 0.25. And if you still want to capture the raw data file, so-called PeakForce capture file, where all the force distance curves are stored, then you need to decrease this value because the system requires that 4 taps done on each place and 1 force distance curve is then saved at this position. And we have the last question here. How is the process different when measuring magnetic forces with the cell, within a cell?
I didn't catch the question. Can you repeat? The magnetic force?
So how is the process different when measuring magnetic forces within a cell? Yeah.
I didn't measure any magnetic force.
Yeah, probably we're not the expert of magnetic AFM here.
Well, I can just comment that in terms if you're talking about the magnetic force measurements, so here in that technique, it's not really in physical contact with the surface. So when we sense the magnetic field or magnetic forces between the tip and the sample, also magnetically coated probe is required. In that case, we've chemically functionalized the AFM probe, and then we bring it into physical contact with this sample surface, also intermittently, as was mentioned today several times, and then we probe and search for the specific interaction between the tip and the sample. And I think with that we've done with all the questions for today. I would like to thank again all the attendants for being with us. And remind you, please take a little bit of your time and fill the survey after the webinar will be ended. And I would like again to thanks very much to our guests today, Dr. David Elstins and Moritz Freundschuh from ETH from Zurich, who are based in Basel in Switzerland. Thank you very much to everybody.
You're very welcome. Yeah. Thank you.
Thank you.